Everyone, good evening. My name is Kajal Kachawa. I'm PMRF Research Scholar from IIT BHU Varanasi. I will be your teaching assistant for the course Host-Pathogen Interaction, that is Immunology, from January to April. And today we are at week 9 of this particular course series. And until week 8, we have covered all the assignments which are available on YouTube. So if you have not visited yet, you can check it out on the YouTube channel or the links provided from the NPTEL. My class timing is every Saturday evening 3 to 5 p.m. So let's start today's week 9. In this today's course, we will be solving the assignment for week 8, that is about antigen and antibodies, and we will also cover T and B cell response. So, lecture material till week 10 is available on NPTEL from Professor Himanshu Kumar. You can check it out from there. In last class, we have studied about the complement pathways. There were three different type of complement pathways. First one was lectin pathway. Second one was alternative pathway, and third one was classical pathway. So in last class, we have seen the three type of complement pathways, and we have also studied about innate and adaptive immunity. Then we have seen a little bit about the antibody structure and function. And we just started about studying antigen and heptane. So today we will continue from that. So if you have any doubt from the last class or last week assignment, you can ask me now. If you have any doubt or any query from the last week assignment or any topic from last week, so we can discuss that first, then we will proceed to today's lecture. Okay, if no doubt, then we will start about what is the difference between immunogenicity and antigenicity. Like there are two different terms. First is immunogen, second one is antigen. So what is difference between them? Like these both are foreign molecules which are recognized by our immune system. Like this immunogenicity and antigenicity are key properties of antigens, but they are different. They are not exactly same. The term immunogenicity, as we can see, it induce immune response, such as antibody production. So the immunogenicity is the ability of a substance to induce an immune response, including the antibody production. Whereas the antigenicity, it is the ability of a substance to be recognized by the immune system, but it may or may not induce an immune response. Like not necessary that that an immune system will be induced or an immune response will be generated. It is not necessary, but it can induce an immune response if it is appropriately modified. For example, with the heptens. So what are these heptens? These heptens are two small molecules. Like they alone cannot generate an immune response, so they lack the immunogenicity. And a heptane, if it is coupled to a carrier molecule, that it can be a protein, then it can induce the immune response. So alone the heptane is very much small and it is non-immunogenic, that it will not induce an immune response. So this heptane needs to be conjugated with the carrier molecule so that it can form an heptane carrier conjugate and it can induce the immune response. So like this we have seen in our last class that there is a experiment. In this experiment what is happening here, like when this small molecule heptane was directly injected into the animal model and it was tested, then there was no antiserum produced. Like there was no immune response against this. But when this heptane was conjugated with the cross linker and the immunogenic barrier, like this heptane conjugate was formed and then it was injected into the animal model, then there was an immune response. There was antiserum was detected in the serum of this animal. So from this experiment we can see that heptane alone cannot induce an immune response, and when it is conjugated to a carrier molecule it can induce an immune response. So alone heptane is non-immunogenic. There are two terms. First is antigen and other one is immunogen. Both sound similar. People get confused between them and use them interchangeably, but these two terms are different. They are not same. Like first we will see about antigen. Like it is a substance specifically bind to antibodies or a cell surface receptor of B cell or T cell. Whereas the immunogen is an antigen capable of inducing an immune response. Like this antigen will bind specifically to antibodies on the surface of B cell or T cell. But this immunogen is an antigen which is capable of inducing an immune response. This antigen can be either immunogenic or non immunogenic, but this immunogen is always immunogenic. And we can say that not all are immunogens, like all antigens are not immunogens, whereas all immunogens are antigens. And this antigen can be either any protein, polysaccharide, lipid, or nucleic acid, whereas this immunogen are normally proteins or large polysaccharides. They are not the like lipids or nucleic acid anything. Immunogens are very specific. These heptanes are low molecular weight molecules which binds to the antibodies. Whereas this heptane become immunogenic when binding to large carrier molecules. So we can say that all immunogens are antigen, but not all antigens are immunogen. You should remember this line. So we were discussing about antigen and immunogen. So we can say that all immunogens are antigens, but not all antigens are immunogens. Like this is very important line. All immunogens are antigens, but not all antigens are immunogens. These heptanes are small molecules that can bind to antibodies but cannot by themselves induce an immune response. Like these are very small that they lack the immunogenicity by themselves. They, for being immunogen, they needs any carrier molecule to be bound with so that they can generate an immune response. So you should keep in mind this concept that all immunogens are antigens, but not all antigens are immunogens. How? Because it depends on who can induce an immune response and who cannot induce an immune response. Like immunogens will always produce an immune response and the antibody production, but all antigens are not capable of producing an immune response all the time. Next concept is a difference between antigens and heptanes. These antigens are toxic or any other foreign substance which induce an immune response in the body, especially the production of antibody. Like these antigens will induce the immune response. But these heptanes are very small molecules which when combined with large carriers such as proteins, then only they can elicit the production of antibodies. They alone cannot induce an immune response. And these antigens are the complete molecule. But these heptanes are incomplete antigens. They will always require a carrier molecule to become an antigen or to generate an immune response. These antigens can bind to the MHC complex. These heptanes cannot bind to the MHC complex. For MHC, we will see the details in upcoming slides. And these antigens can directly bind to antibodies, but these heptanes cannot bind to antibodies directly by themselves. And these antigens can trigger an immune response by themselves. Keep in mind, by themselves. But these heptanes cannot trigger an immune response by themselves. They need a carrier molecule. Like heptanes are of no point without a carrier molecule. So remember this difference between antigen and heptanes. And the first we have seen difference between antigen and immunogen. So there, I think we have you all have gone through the lecture slides. So I will quickly summarize and cover those lecture slides for you. And if you have any specific doubt, you can stop me in between so that we can discuss, or you can drop a comment later. So, the properties of immunogens which contribute to the immunogenicity are like their foreignness. They are foreign molecules, they are not of our own body. And their molecular size, like how large, how big. Yeah, so we were discussing about the properties of immunogens that contribute to the immunogenicity, that is the induction of an immune response. So first one is their foreignness, like they are outside material for the body, they are not body's own cells. And their molecular size and their chemical composition and complexity, their ability to process and present antigen or immunogens on an MHC. So these are the properties that are responsible for generating an immune response. Yeah, so we were discussing about the properties of immunogen, which contributes to the immunogenicity. It got stuck in between, so I'm so sorry for that. We'll start this portion again. Like this, immunogen should be foreign. It should have a particular molecular size. And its chemical composition and complexity also matters to induce an immune response. Its ability to process and present antigen or immunogen on MHC cells, MHC molecules, which we will be studying in upcoming slides. So the first property is its foreignness. First property is the foreignness. The immunity of the host do not develop immune response to self antigens. So the degree of its immunogenicity depends on the degree of its foreignness. Like immunogenicity requires a molecule to be foreign. It will not develop an immune response to the self antigens. Like for example, like this bovine serum albumin, which is BSA, it is not immunogenic when it is injected into cow, or it is less immunogenic in goat. But it is when injected into chicken, it is immunogenic. Why? Because for host, this is bovine serum albumin is taken from the cow. So it is not immunogenic in cow. And in goat, it is less immunogenic because of the similar nature. But when it is injected into chicken, it is highly immunogenic because of the difference of the host. And for chicken, it is highly foreign. So its degree of immunogenicity depends on degree of its foreignness. Unlike some macromolecules are highly conserved throughout evolution and display little immunogenicity, like for example, cytochrome C or collagen And like for some exceptions, this corneal tissue and sperm, they will be immunogenic. So this is another example of foreignness. And the next property of immunogen is its molecular size. Like the active immunogens or the good immunogens should have a size range of more than this much Dalton. But if it is less than 5,000 to 10,000 Dalton, then it will be poor immunogenic or less immunogenic. But more than this much Dalton, it will be highly immunogenic. So there is a requirement for molecular size. Like here is a list given for the antigen and its approximate molecular size in Dalton. So you can check it out from the slides. The third property is its chemical composition and the complexity. Like for a protein molecule, there are four organizational levels, like four structural levels. The first one is, as we know, I think you all have studied this in very basic biology, but I will summarize it here. Like the first is primary structure, which is a linear chain of this amino acid sequence. So this primary structure is a linear chain of amino acid or the polypeptide chain. The second level of organization is secondary structure, where it start forming alpha helix or beta pleated sheets. And the third organizational level is the tertiary structure, where it start folding. Like there will be like this alpha helix and beta sheets, they will interact with each other and they will start folding into a structure by the use of some bonds like hydrogen bonds and disulfide bonds. Then it comes the quaternary structure, which is dimeric protein molecule, like two different subunits, like two different tertiary structures. They will combine together, like two different subunits can combine together and form the quaternary structure. So this chemical composition and complexity of the immunogen is also determining the level of immunogenicity caused in the host cell. So if it have primary structure, the immunogenicity will be different. For secondary, tertiary, and quaternary, the immunogenicity level will be different. Like this polymers composed of multiple copies of same amino acid or sugar tend to be poor immunogens. And the lipids are the heptanes, and they need to be conjugated with carrier molecule to produce antibodies. These are important for the assays like for detection of steroids and vitamins. So the chemical composition also matters for immunogenicity. So the next we will study about the susceptibility to antigen processing. So anybody would like to comment on this? Like what is antigen processing? So, what happens in the case of antigen processing? The large insoluble macromolecules are more likely to be phagocytosed for processing. So, what happens here? Like the nature of antigen influence its processing and its presentation by the immune system. And these antigens are presented by MHC. MHC have two classes, MHC class 1 and MHC class 2. What are these MHC? These MHC are major histocompatibility. complex. These are MHC. There are two type of MHC, class 1 and class 2. These MHC molecules are responsible for antigen presentation to the immune cells so that immune cells can recognize them. So there are like two pathways of antigen processing. First one is MHC class 1 pathways. Second one is MHC class 2 pathways. This class 1 pathway is also known as the endogenous pathway. And this class 2 pathway is also known as the exogenous pathway. So this MHC class 1, it processes the intracellular antigens like viral proteins or the tumor antigens. And like when the cytosolic proteins are degraded by the proteasome, then the peptides enter into the ER or TAP transporter. Then they are loaded onto this MHC class 1 and they are transported to the cell surface, and then they are presented to cytotoxic T cells, which are CD8 and CD4 positive, that we will discuss in upcoming slides. So basically what happens in this, initially, like the cytosolic proteins are degraded by the proteasome here. Then these peptides will enter into the ER and via this TAP transporter, and then these will be loaded onto the MHC class 1 molecules and transported to the cell surface here. Whereas in the case of MHC class molecule, what happens, like they will process the extracellular antigens. Like they will take the extracellular antigens and like bacterial components or any soluble proteins. So like this, when this antigen is engulfed into endosome and they are degraded, then MHC class 2 is synthesized in the ER and it is bound to an invariant chain for blocking the premature peptide binding. And here in this M2C compartment, what happens? This invariant chain is removed and this peptide is loaded. And this will be presented on again the cell surface. In the case of this MHC class 1, the antigen is presented to CD8 positive cells. Whereas in the case of this MHC class 2 molecule, the antigen is presented to the CD4 positive cells. So there is a difference for MHC class 1 and class 2, that this class 1 will process the endogenous this antigens like any tumor or viral antigen, whereas class 2 will process the exogenous antigens, that is any bacterial component or any soluble protein, and these are the cells to which they will present the antigen. So the next is the role of biological system in immunogenicity. So the first one is host genetic makeup. Like each individual has a unique MHC profile, and some antigen are better presented by specific molecules. So the host genetic makeup also matters here. Like the say one antigen can be immunogenic in one person but not in the other because of the difference in its genetic makeup. And the second one is here the route and amount of this challenging antigen. Like the route of administration also matters here. If route of administration is like if it is subcutaneous or intramuscular, so it depends on that. And the third one is the use of agents or any adjuvants to enhance the immunogenicity. So these adjuvants or any agents are the substance added to vaccines to enhance their immunogenicity. So we will take this one by one. The first one is host genetic makeup. Like every host has a unique genetic profile, which means their immune system recognize and process antigens differently. And this variation can lead to the different immune response to the same antigen across two different individuals or two different species. So this genetic makeup of recipient host and the genes of MHC control response to antigens. Like what happens in this case is MHC genes are central to immune recognition, and this MHC molecules present antigenic peptides to T cells. So the polymeric nature of this MHC will determine which peptides can be presented. So the some individuals may not respond to certain antigens simply because their MHC does not bind to those peptides efficiently. The next property is genes involved in coding for antibodies. These include genes for the variable region of antibodies that we will study in upcoming slides. The next one is B cell and T cell receptor genes. Like their genetic basis also decides the diversity and specificity of immune recognition by the host. And the next property is route and amount in which the material is presented. Like if it is too low or it is too high, then it can induce a tolerance. Like there should be a specific route or specific amount. Like if it is too low, then the immune system will ignore the antigen, which is called the immune tolerance. And if it is too high, then it can trigger the tolerance or the immune suppression instead of the activation. And the single dose is sometimes not enough, so a booster dose is needed. Usually, one exposure to the antigen does not generate strong memory. So that's why we need booster doses to again induce the memory so that it can be a long-term immunity. So, like what happens here if this dose is too low, then what will happen? Immune system will not recognize the antigen and it will cause the immune tolerance, and this antigen will not be recognized. The second case is if it is too high, then what will happen? It will trigger the tolerance, or it can also cause the immune suppression. So that's why specific dose is required, specific amount is required. The next one is the route. If it is given intravenous, that is the into the vein. If it is given intradermal, into the skin. If it is given subcutaneous, that is beneath the skin. If it is given intramuscular, that is into the muscles. If it is given into the intraperitoneal, that is into the peritoneal cavity. So it will depend on what route of administration we have used. Now, next one is epitopes, and these are the antigenic determinants which are recognized by B cell and T cells. And these B cell epitopes tend to be on the outside of the antigen. For example, the hydrophilic amino acid is on protein's surface, whereas the hydrophobic amino acids are inside the protein. And the T cell epitopes from proteins derived from enzymatic digestion of peptides and then their association into MHC. So these are the properties of the epitopes that you can go through the lecture slides. If you have any doubt, then you can ask. Like there is a difference between antigen recognition by B cell and T cell. There are differences between that. Like the interaction with the antigen, for B cell, it involves the binary complex of membrane Ig and antigen. In the case of T cell, it involves the ternary complex of T cell receptor, antigen, and MHC molecule. And for binding of B cells binding of soluble antigens with B cell, it is possible. In case of T cell, it is not possible. And in the case of B cell, the involvement of MHC molecule is not required, whereas in case of T cell, it is required to display the processed antigen. And in the case of B cell, the nature of antigen can be protein, polysaccharide, or lipid, but in the case of T cell, it is mostly protein, but sometimes it can be lipid or glycolipid. And the epitope properties of B cells are accessible because they are hydrophilic and the mobile peptides containing sequential or non-sequential amino acids. Whereas in the case of T cells, they are internal linear peptides produced by processing of antigen and bound to MHC molecule. So there are differences between how these antigens will be recognized by whether B cell or T cell. Next, this is the workflow for generation of monoclonal and polyclonal antibodies. So let's see how this happens. So in the case of this generation of monoclonal and polyclonal antibody, the workflow used here is like the like first we need to inject the mouse with an antigen, required antigen, which have multiple epitopes. Like it have four different type of epitopes, one, two, three, and four. There are four different type of epitope. We have injected this mouse with the antigen. Then this immune system will generate the B cells, which are specific to each epitope. So there can be two possible outcomes. First is like it can produce the polyclonal serum so that we can isolate directly from the immunized mouse. It will contain a mixture of different antibodies like for different epitope. Antibody 1, 2, 3, 4 for the respective epitope 1, 2, 3, 4. So it is a polyclonal serum. So first we can isolate that, or the second option is we can isolate the spleen cells or the B cells from the mouse. And this isolated B cells 1, 2, and 3, 4 respectively, these will be fused with the myeloma cells, which are immortal. And this will form this their hybridization will form the hybridomas, which contain the myeloma cells, which are immortal, and along with the myeloma cells, it contain B cell 1, 2, 3, 4 respectively. So then we can clone a large quantity of this identical, like we can select these and we can clone into a large quantity of the single epitope, which are antibody 1, 2, 3, 4, which were respectively for the epitopes 1, 2, 3, 4 that we have taken from the initial beginning for injecting the mouse. So there is a case that either it can produce the polyclonal serum, which will contain both, like all the four antibodies, or we can take the monoclonal, or we can produce the monoclonal antibodies by fusing this with the myeloma cells and forming a hybridoma. Then it will produce the four different type of antibodies. So the next one is applications of this monoclonal and polyclonal antibodies. Like this monoclonal antibodies can be used for the basic research like immunoblood, immunohistochemistry, and for flow cytometry applications. It can be used for the diagnosis applications like this pathogen detection, malignancy test, and diagnosis of an array of infections and autoimmune disease. It can also be used for the therapeutic cases like for the cancer treatment, autoimmune disease treatment, and the allergy treatment. And in the case of this polyclonal antibody, these can also be used for like basic for the immunoprecipitation, chromatin immunoprecipitation reaction, and immunoelectrophoresis, different immune tests. And for in the case of diagnosis, it can be used for the histopathological analysis. And for therapeutics, it can be used for the passive immunization or antivenom. So there are applications of this monoclonal and polyclonal antibodies. So from where the concept of this antibody was initially started, like how this the conceptualization of the soluble components or the antibodies were started. We have seen in our very initial classes of the history and development of immunology, there we have seen this experiment from Emil von Behring. So in this experiment, like if you remember from the very first classes, then it's fine. Otherwise, you can check it out from the initial lectures. I will quickly summarize this. Like Emil von Behring have done experiment. He have taken three different groups of this experiment A, B, and C. In the first case, the serum, the guinea pig one was there, and he have it was unexposed to any type of antigen. Then he have taken the serum from this unexposed animal and injected into the guinea pig two. And in the case of the second group, what he have done, this was the first group, it is second group, this is third group. In the second group, he have taken a sublethal dose of toxin from bacterial strain A and injected into this guinea pig 1. Then serum from this animal exposed to toxin A was injected into this guinea pig 2. Then in the third case, he have taken a sublethal dose of toxin B and then he have taken the serum exposed from the then he have taken the serum from exposed animal to the toxin B, then injected into the guinea pig. Then what he have done, he have observed that at last, when he have injected all the animals with a lethal dose of toxin from bacterial strain A, then he observed that the first this guinea pig died because it do not have any type of this anti-sera in its body, so it died. And in the case 3 also this died because it had the antisera for the toxin B, but here it is injected with the bacterial strain A. But only the second case animal live because it was initially exposed with the toxin A. So this was a case, you can check it out in detail from the initial video. So the basic concept from this experiment was that he confirmed the role of antibody in immunity. Like he told that there are some antibody or some sera produced which are responsible for providing immunity to the guinea pigs. The next was the next study was of the toxin neutralization assay by Paul Ehrlich. Just a minute. Yeah, so second experiment regarding this antisera was of Paul Ehrlich, where he have seen that. Oh, sorry. Where he have discussed the case of this toxin and the antitoxin conjugate. Like with the toxin, if we give an antitoxin, then it will cause a neutralization effect and cell will be live. But if only toxin is there, then cell death will occur. So from here, he told that there are the antitoxins are the antibodies that neutralize toxins before they can cause the harm. So this was also experiment from Paul Ehrlich about toxin neutralization assay. You can check it out from the previous videos. So from this, they have given the concept like there are some antibodies in the serum which are responsible for providing the immunity to the cells. So they have told that this serum is a fluid phase that remain after the blood clotting, and these antibodies are also found in other secretions, other bodily fluids. So the molecular composition of an antibody is that these antibodies are heterodimers. They have two light chains and two heavy chains, and the first 110 amino acids of the N-terminal end of heavy chain and light chain will vary. They will be different for each antibody, and they will decide the antibody specificity that we will study in detail in the antibody structure. So here we can see that this is a typical structure of the antibody. Here we can see that it have two light chains and two heavy chains, and it have the upper portion, that is the fab, that is the fragment of antigen binding, and it have the lower, this fc region, which is fragment of crystallization, which have this effector activity or the biological activity, and this fab region or the antigen binding region, it have the cdrs, that is the complementarity determining regions, which are which are responsible for determining the type of antigen it will bind to. And this fc region have the effector activity or the biological activity, which will tell about the opsonization or the complement activation or the ADCC activity, that is the antibody dependent cell mediated cytotoxicity for killing the cells. And it will also tell about the soluble antigens which will bind to antibody and form immune complex. So like it have two regions. First one is this fab region, which is fragment of antigen binding. This fab region contain the CDR region, which are responsible for binding to antigen. And the second one is this FC, which is fragment of crystallization. This FC region have the effector activity. This effector activity is nothing but the biological activity, like how it will respond to the antigens. So like this have two regions, Fab and FC. Then it have the light chain, sorry, light chain and the heavy chains. It have two light chain and two heavy chains. And this light chain are 214 amino acids and this heavy chains are 446 amino acids. And it also contain the disulfide bonds, which can be intra-chain disulfide bonds or it can be the inter-chain disulfide bonds. So the number of disulfide bonds are different in different classes of antibody that we will see in the upcoming slides. It have this N terminal, which is the amino terminal responsible for antigen binding. Then it have the C terminal, which is responsible for binding to the immune cells. So this is the structure of an antibody. Like they have also digested the antibody, like they have removed these disulfide bonds, then they have digested this from here central part with the this papain enzyme, which are responsible for digesting this above the hinge region. Hinge region is nothing but the central region of an antibody. So when they digested this with the pepsin enzyme, they observed that there are different fragments of antibody. That is because this pepsin enzyme cuts below the hinge region. That is, it cuts here. So it have this upper portion and the lower chains of the antibody. And when they digested this with the papain enzyme, it cuts the above the hinge region. So they have different chains of antibody. Then when this digested antibody was run onto a gel, Then they have observed the different antibodies, like different chains of the antibody, different fragments of the antibody. From there, they confirm that yes, there are the FAB region, FC region, and this different light chain and heavy chain are observed on the gel. So this was experiment by using enzymatic digestion. So there are this we have just discussed about light chain and the heavy chain. There are this amino terminal end have 110 amino acids which are variable, which are responsible for binding to the antigen. In light chain, it can be either kappa or the lambda. In human, the 60% is kappa and 40% is lambda, whereas in the case of mouse, it is 95% kappa and 5% only is lambda. So these are two type of light chain. And for the heavy chain, there are like five types of this heavy chains for different antibody, which determine the type of antibody. So, like it can be IgM, IgG, IgA, IgD, or IgE. Each class of antibody have a different heavy chain. It can be this mu, gamma, alpha, delta, and epsilon. Stain composition of the five immunoglobulin classes in humans. So you can check it out from the lecture video. Like there are five classes of this antibody in the human chain, and it have different heavy chains, and these heavy chains also have the subclasses, and the light chain can be either kappa or lambda in human, but majorly this is kappa. And depending on the type of this subclass of heavy chain and the light chain, there is a molecular formula for each antibody, like what type of antibody will be there. So there is a hinge region that I just talked about. This hinge region is the central part here. This is the hinge region here. So this is rich in proline and cysteine, as it gives the flexibility to the antibody. So there are five subclasses of antibodies. First one is IgG, then IgD, then IgE, IgA, and IgM that we have seen in the last class. So I will take a quick revision of this. Like this IgM is a pentameric structure. Like there are five molecules of IgM are present. Then they form a complete pentameric structure. So this IgM is the first antibody produced in response to the microbial attack by the B cells, and it is the largest antibody, and it is found in the pentameric form. It circulates in the blood and lymph and constitutes 6% of the total antibody content in the serum. And it involved the agglutination and the opsonization. It has a large number of antigenic sites on its surface because it is pentameric, and therefore it facilitates the efficient activation of the immune system. So these points about each antibody you have to keep in mind. The next is IgE that we have been studying from the initial classes, that it is involved in the allergic reactions, and it is also involved in the parasitic infection. So there are the cells like this mast cells which contain the granular structure which have this histamine and the allergic, which are responsible for allergic reactions. So when an antigen is bound to this antibody, this will cause the degranulation and the release of the granular content, which will release the antihistamine molecules. So this IgE is present in the least amount. This is very less. It only about 0.02% in the total antibody content. And these are present in the linings of the respiratory and intestinal tracts, and they respond to the allergic reactions. And this is found as a monomer in the body, and it weighs about uh, this much daltons. The next one is IgD, which is membrane bound on the B cells. These antibodies can be either membrane bound or they can be secretory. So the case of IgD, it is involved in the production of antibody by B cells. It is present as a monomer and weighs about this much Dalton. It comprises less than 1% of the total antibody content, and it acts as a receptor on B cell, and it participates in the B cell activation and differentiation. This next one is IgG. It is the most abundant antibody. So this is the most abundant isotype in the plasma, and it comprises 80% of the total antibody content in the serum. And it can detoxify the substance that are harmful, and it recognizes the antigen antibody complex. It is transferred to the placenta through the fetus and protects the infant until its birth. Like before birth, IgG is present in the body, and it is divided into four subclasses, which one is IgG1, G2, G3, and G4. Among these, only IgG3 and G4 possess the ability to cross the placenta. And the heavy chains of IgG have two antigen binding sites and are of subclass gamma, and it facilitates the process of phagocytosis, and it provides the immunity to the developing fetus. It can neutralize the toxins and pathogens, and it offers protection to the body. Now last one is IgA, which is found in the dimeric form. It is a secretory antibody. It can cross-link large antigens. So it is usually found in liquids such as the breast milk, serum, saliva, and fluids of the intestine. IgA in the breast milk, it protects the infant gastrointestinal tract from microbial activity. It constitutes of 13% of the total antibody content, and it is divided into two subclasses, which is IgA1 and A2. And among these, IgA1 is highly found in secretions, and it is also called as the secretory immunoglobulin. It exists in both monomeric as well as dimeric form. It provides the first line of defense. Keep in mind about this first line of defense against the pathogens and limits the inflammation. It also activates the complement pathway and participates in the immune response. So this is a comparison table of all the four, like all the five type of antibodies along with their subclasses. Like this, IgG is divided into four subclasses, and IgA is divided into two subclasses. Other three are single classes. So their molecular weight, their heavy chain component, their normal serum level, their in vivo half life, and their ability to activate the complement pathway, that either they can cross the placenta or not, and their presence on membrane of mature B cells, and whether they can bind to Fc receptor for phagocytosis, their mucosal transport, and they can induce the mast cell degranulation or not. Their properties are mentioned here, so you can check it out from here. Like this, IgG have the longest half-life in the serum. So IgG1, G2, and G4 have the longest half-life. And only IgG is responsible for inducing mast cell degranulation. And only this IgG can cross the placenta. So there are these different properties of all the antibodies. You can check it out from the table. And the slide chains can be of kappa and lambda that we have just seen. And there is a other thing about this antibody, about the VTJ recombination and the glass switching, which is a detailed topic. So I will just quickly summarize it here. If you have any doubt, then you can let me know so that I can cover it in the next class as well. So there is a rearrangement of this heavy chain and the light chain, and there is a generation of this antibody diversity by the VDJ joining and the recombination and rearrangement. So I will just quickly summarize like what is happening here in this case. I will give you the brief concept and the overview of what is happening and why is this happening. I will not go into the detail. So this VDJ recombination is a unique genetic process that generates the enormous diversity of antibodies in the B cell so that we can like our immune system can respond to millions of different type of antigens. I will write a few things here. V stands for variable segment, D stands for diversity segment, J stand for joining segment or region, you can say it anything. So it happens in the bone marrow during early B cell development. So for heavy chain, what will happen? Like in the case of heavy chain rearrangement and light chain rearrangement, the case is different. In the case of heavy chain, this 1D segment will join with the J segment. Then this V segment will come and join the DJ combination. And it will form a VDJ unit. And it will code for the variable region of the antibody. Variable region where antigen will bind. And in the case of this light chain, D segment is not present. So only this V segment will join with the J. There will be only VJ. It will also code for the variable region. Like here it will code for the heavy chain, here it will code for the light chain. Like this will create the antibody diversity before an antigen exposure so that we can have a different type of antibody produced for a different type of antigen that we encounter. So like why this is required, why we are talking about this VDJ recombination? Because our body encounters millions of different type of pathogen in our lifetime. So for each pathogen that will have a unique antigen that our antibody will recognize. But we cannot store like these millions of different types of antibody in our body. So what we do like Yeah. So what we do, like instead of having a separate gene for each antibody, we use this VDJ recombination so that these segments can be joined randomly, that can create a new combination so that we can have a different type of antibody for each type of antigen. So this is the basic mechanism of why we are having this different type of combinations for heavy chain and light chain. Let's suppose, for example, we have 40 types of V segments. We have 25 type of D segments. We have 6 different type of this J segments. Then how many different combination of heavy chain we can have from this VDJ? All three are there, means it is for heavy chain. If only V and J are there, means it is for light chain. So in case of this heavy chain, like we can have 40 into 25 into 6 is equal to this much combinations of the heavy chain. And same in the case of light chain. Like for light chain, we, like suppose we have this 30V and this 15J. Then how much we have this 30 into 15 is equal to this much combination. So overall we will have this, by combining both the combinations, we will have millions of antibody combinations possible, which are responsible for binding to millions of different antigens. So this is the VDJ combination. So these are light chain and heavy chain. How this can be rearranged? They can be bind randomly and they can form a different type of polypeptide chain. So that you can check it out from the lecture video. Now the next topic is MHC and T cell receptor. As I have already told you, the MHC are the major histocompatibility complex, and these T cells are present on the immune cells. These are the receptors. So this T cell receptor is a molecule. It is found on the surface of T lymphocytes or the T cells, and it allows the T cells to recognize and bind to antigen peptides, but only when these peptides are presented by MHC molecule or the antigen presenting cells. Like they themselves cannot go and bind to the antigens. So what happens here? This MHC class 1, as I have already told you in the previous slides, that this MHC class 1 will bind to, sorry, this MHC class 1 will bind to CD8. Sorry, I think that is out of the space. MHC class 1 will bind to CD8 positive cells, and the MHC class 2 will bind to the CD4 positive cells. Like this is here antigen presenting cells, and the MHC class 2 molecule is present on the surface, and they have captured a peptide or it can be a antigen. So this is here the T cell receptor. This is T cell and this is the T cell receptor. So this MHC class 2 have taken this bound or the captured antigen to the T cell receptor. And here the CD4 is present because the MHC class 2 is presenting the antigen. So it is presenting antigen to the T cells which have CD4 on its surface. If it was MHC class 1, then it might be here CD8. So that is the difference. So this is the structure of the immunoglobulin and the T cell receptors. The structure of immunoglobulin or the antibody we have just seen. And the structure of this T cell, it also have this variable and constant region which have alpha and beta portions, and there is a connecting sequence, and there is a transmembrane region, and the cytoplasmic tail, which is the C terminal here, and here this is the N terminal. So this is the structure of the T cell receptor, and this is your immunoglobulin. So this is the structure for your CD8 molecule, and this is for this is for CD4 and CD8 molecules which are present on your T cells. So there is a list, you can check it out from the presentation. Like on the CD4 positive T cells, like there are so many different type of receptors present or the interleukins produced. So you can check it out from the slides. So then here it's antigen and antibody topic ends, and then starts the pathogenesis part, which is viral pathogenesis. So what happened in the case of viral pathogenesis? Like this, what is pathogenesis? Pathogenesis is like basically physiological changes caused by viral replication, which leads to the symptoms. Like these are the changes in the physiology, which results to the deviation of vitals or some changes in the basic body functioning, and it culminates to the clinical symptoms due to viral replication. This infection could be productive, abortive, latent, or reactivation. So, what are these different type of infections? Like this infection can be acute infection where the onset is rapid and it is for short duration, and like it can be latent infection where the this virus lies dominant for a longer time after the initial infection. Then it can reactivate multiple times, like there will not a single time for infection. It can be latent, like it can, after some time, it can reactivate again and again. This can be progressive, like after an initial infection, viral load slowly increases over time. Like this will keep increasing, and eventually it will lead to severe disease or the immune system failure. It can be chronic or persistent infection, like this virus is never fully cleared from the body. There will be continuous low level of replication occurring in the virus in the body. So it can be chronic or persistent for a longer time. And then there can be a failure to clear all evidence of infection. Like there can be incomplete clearance. Like it can be acute infection will resolve, but viral nucleic acids will persist there. So it is in unclear whether it is have any evidence left or not. So it can be incomplete infection also there. This virus infection and disease outcome, what we can see from here that like we know that some individuals get severely sick while others barely show any symptom, or some will show no symptoms at all. Why this happens, different type of outcome in different individual. Like depending on the virus species or the host, and the depending on the host species, like on which the infection is happening, or the individual host variation, like its genetics or the immune system or the microbiome of an organism that will decide whether the infection will be severe or less severe or no infection at all. Like not a single virus can cause same type of effect in all the different type of individual. That will be different for all individuals. So in this experiment, what you can see here, there is a quasi species and bottleneck in tissue tropism. So what are these quasi species? These are the viral populations which don't exist as one uniform strain. Instead, they form a cloud of variants called the quasi species due to high mutation rate. Like they will not be same for all the upcoming infections. They will keep changing their variant. And this bottleneck is the restrictive step where only a small number of virus can manage to pass through and seed infection in the next tissue or the next cycle. Not all type of virus can pass through this. Like in the quasi species, we can see here this multiple type of variants can coexist. Different type of virus variants can coexist. And when it comes to bottleneck or infecting the second tissue, what happens? Only a certain specific type can pass through and cause infection in the next tissue as well. So here in this case, what is happening? Like different individuals will respond differently to the same virus. Here the virus is of particular type. Virus is of particular type. And when this virus infect to the host, it will go into the host, then host gene 1 will respond to this virus differently. And when this virus will go into the next host, then again the subsequent host, all these different type of host will have different type of outcome. It will not be same because the virus is it keeps on mutating, it keeps on forming the different variant, so the outcome is different for all the cases. And why these variants are different for all the different hosts? Because of their the cytokine networks, their epigenetic factors and the viral factors, and also depends on the timing for various infections and the difference events. The features of the pathogens or the virus. Like these can survive and multiply in the appropriate host, and how it is possible? It finds a nutritionally compatible niche in the host and it replicates using the host resources. Then it will colonize inside the host and it will avoid and subvert or circumvent the host innate and adaptive immunity. Then it will cause the pathogenesis. So now let's start the week 8 questions, the assignments for week 8. I hope you all might have submitted this because the deadline is already over on 19th of March. The first question was that what role do MHC molecules play in the immune response? So we have seen that they process and present the antigens to the T cells. So this is the main function of the MHC molecules. We have also seen that I hope you now remember this MHC1 will present antigen to the CD8 positive cells and this MHC2 will present cell antigen to the CD4 positive cells. And this MHC1 will process the endogenous antigen, whereas this MHC2 will process the exogenous antigen. These points you have to keep in mind regarding MHC. This we have seen in the initial slides. The second question was, which of the following accurately describes IgM? So we have seen the properties of this IgM, that it is the first antibody produced in response to an antigen. It exists only in the monomeric form. No, it is a pentameric form. IgM has the highest complement activation potential. IgM is the most abundant immunoglobulin in the serum. No, this is IgG is the most abundant immunoglobulin. IgM is present in monomeric form on mature B cells. Yes, on mature B cells it is present in monomeric form. And this IgM can also be found in serum in a pentameric form. Yes, it can be both. IgM has the highest half-life. No, IgG has the highest half-life. IgM does not bind complement proteins. No, it binds to the complement proteins. So these are the properties of the IgM. The third question was, in the context of immunoglobulin diversity, which enzymes are essential for initiating VDJ recombination in B cells? So for this, we have this enzyme called RAG1 and RAG2. The name of these enzymes are recombination activating genes. These are the initiators of the VDJ1 and VDJ2. These two enzymes. Without these two enzymes, recombination cannot start. This RAG1 and RAG2, they stands for the recombination activating genes 1 and 2. These enzymes are only expressed in developing lymphocytes, and their main function is to recognize recombination signal sequence, which are RRR. They will recognize these sequence that flank the VDJ gene segments, and they will introduce the double stranded breaks between the coding segment and the RSS. Then they will bring together the correct gene segments for joining, and without this RAG1 and RAG2, recombination cannot occur. So no functional B or T cells will be produced. So these are the initiators of the BDJ recombination. The next question was, which T cell subtype plays a significant role in inflammation and is also linked to autoimmune disease promotion? So the correct answer is TH17. And we have seen from the slide that this TH17 is responsible for playing a significant role in inflammation. Yes, we can see here this TH17, it induces inflammation and it promotes the autoimmune disease. Then the next question was, which term describes a viral population within a host that consists of many genetically distinct variants due to high mutation rate? These are the quasi-species that we have seen. There are multiple variants of viruses existing together. So these quasi-species are that. The next question was, heptanes require a larger carrier protein to elicit an immune response because these heptanes are too small to be immunogenic by themselves. So they alone themselves cannot generate an immune response. So they require large carrier proteins to elicit an immune response. This we have been seeing from the last two classes. Our next question is, what is the significance of kappa and lambda chain in antibody? So we know that this kappa and lambda chain in the light chain. These contribute to the genetic diversity in immune response, with kappa being predominant in human. Yes, we have seen this. This kappa is about, in the case of humans, in light chain, this kappa is about 60%, and this lambda is about 40%. The next question was, when is IgM produced during an immune response and what is its primary function? So we are seeing the detail of IgM from so many slides that this IgM is the first antibody produced during the primary immune response and is essential for early immune defense. So let's check out the other options, like this IgM is the first antibody produced during secondary immune response. No, this is produced in the primary immune response. This IgM is produced later during an immune response and is responsible for allergic reactions. No, IgE is responsible for allergic reaction. This IgM is only involved in autoimmune disease. No, there are other roles also. This IgM is produced solely for parasitic infection. No, it is produced in all type of infections. In the structure of antibodies, which component is directly responsible for antigen binding? Very direct and very simple. The answer is CDR, that is complementarity determining region, which is the variable region of the light chain. So the other options are this FC region. No, it will bind to the immune cells. The constant heavy chain region. No, the constant region is downside, the antibody at the lower side, which is in the FC region. The light chain constant region. No, the light chain heavy region is responsible. Sorry, light chain variable region is responsible for binding to the antigen. Glycosylation sites, no, these are the sugar molecule site present on the antibody. So they have no role in binding to the antigen. The last question was, which of the following statements about antigen and immunogens are correct? So all antigens are immunogens? No, all immunogens are antigens. Immunogens can induce an immune response, but not all antigens can. Yes, antigens are always immunogenic, not always immunogenic. Immunogens are always recognized by the immune cells. Yes. Antigens may or may not trigger an immune response. Yes, absolutely correct. Immunogens are involved in triggering antibody production. Yes, they can produce antibody. Antigens are always harmful to the host. No, they are not always harmful. Only immunogens are used in vaccine development. Yes, because they will generate an immune response for sure. So these were the questions from the last week. And this is the summary of antigen and immunogen. Like all immunogens are antigens, but not all antigens are immunogens. Just mug up this line for the immunology. So till now, these topics are covered already on the NPTEL channel. So you can check out their lecture material and the assignments. If you have any doubt or any questions or any queries, you can post it in the discussion forum of the NPTEL, or you can leave a comment down the video, or you can put an email to me, or we can discuss it in the next classes. So that's all for today. Thank you everyone.